Hello, my name is Erin Lee. I am a sophomore bioengineering major and sustainability minor in the Science and Global Change CP Scholars program. Over the summer, I worked at the Institute for Bioscience and Biotechnology Research in order to confirm NTIR gene mutations in Nicotiana glutinosa. My lab's goal is to see how mutations in the NTIR gene of Nicotiana glutinosa affect the plant's immune system, otherwise known as the hypersensitive response. By engineering a more stable NTIR domain, the hypersensitive response will be broader and more sensitive. These findings can then be applied to other plants, such as food crops, to increase their immunity to disease. In order to carry out my research, I acquired many new techniques, as you can see in the Materials and Methods section. Some of the main techniques I used were amplifying the DNA that had the mutated fragment with PCR, isolating the desired fragment of DNA with a restriction enzyme digest, assembling the desired mutation in another DNA fragment with Gibson's assembly, transforming the DNA into new E. coli cells with electroporation, and confirming that the DNA sequence of the cultured cells matched what was expected with another digest, as well as checking the sequences with APE software. In the future, the DNA that I sequenced will be transformed into agrobacteria cells to then be applied to Nicotiana glutinosa so we can physically see any changes to the plant's immune behavior. I learned a lot of plant biology, as well as bacterial biology, from this project, and how meticulous lab work can be. I also learned how important it is to keep track of experiments down to the minute detail, in case the detail is needed in the future, or if I forget what I did the first time around. If you're interested in this topic, my advice would be to not be afraid to reach out to professors who study or research plant science, because they will always welcome curious students who want to learn more about their work. Thank you for listening.